Good morning, everyone. Welcome to the Paase webinar series. Um, Today, we have a speaker from the Philippine Genome Center. And he's currently the director of the DNA sequencing core facility. He has a PhD in applied marine biotechnology, leveraging the current molecular toolbox in the practice of disease management in aquaculture, and of course now of all different types of disease models. In fact, in 2017, he has started developing some technology that analyze or detect white spot syndrome in diseases such as shrimp, and then also have been using several high throughput sequencing platforms. He has been the director of PGC, especially during this time with COVID. And so he has been doing a lot of sequencing of our COVID from the population. In fact, they have identified several strains that are seen here in the Philippines, and it has been very important for our national uh, crisis right at the moment. Currently, today he will be talking about some of the sequencing efforts that they have been doing in the Philippine Genome Center. And we're very excited to see that some of the other services that maybe they are offering as well. So they're just not focused on COVID, but also other researchers, maybe like cancer, if you're interested in that, or even environmental samples. So without further ado, I would like to welcome Dr. Benedict Maralin.
Hello po, good morning sa lahat. First of all, I'd like to thank Sir Mike for inviting me to talk to this Paase webinar. Okay, so let me share my screen to start. Okay, I hope everyone can see it now. Good morning, everyone. Thank you for attending to
Well, looks like we have some technical difficulties.
Opo. Sige po, wait lang po.
Yeah.
I think I'm ready na. Sorry for the interruption. Good morning, everyone. Thank you for attending today's Paase webinar. You can call me Ben, and for this presentation, I'd like to talk about the SARS-CoV-2 sequencing activities in the DNA sequencing core facility of the Philippine Genome Center. We at the Philippine Genome Center are composed of diverse personalities from our internal administrative services to our stakeholder engagement personnel. However, one thing is common to all of us. We are passionate at everything that we do, and as I introduce you to our facility, we hope that together we can leverage genomics for a better Philippines. The DNA Sequencing Core Facility, or DSEF, is but one of the different core facilities in PGC. Most of our research programs are implemented with the DNA sequencing services offered at DSEF. Established in 2011 through cumulative grants from the OSTP Shared and CHED PICARI programs, we are one of the first facilities to be established in PGC that's really service-oriented and catered to not only researches being done in the University of the Philippines, but also to our scientist colleagues in different universities in the Philippines. Actually, PGC's reason for existence is responding to pandemics such as COVID-19. 10 years ago, when there was a SARS scare, we have been envisioned to lead in the country's genomic surveillance effort to manage highly relevant diseases that lead to pandemics. So here we are fulfilling that vision while also serving as the only local sequencing facility in the Philippines. Since 2011, we have been serving more than 566 regular clients in their molecular biology and sequencing needs. As mentioned, we have also been directly involved in our country's COVID-19 support through testing and developing our molecular testing program. In general, we have three major activities in the DNA sequencing core facility of the Philippine Genome Center. These are research, training, and provision of access to several genomics tools. Here is a general overview of the diverse genomics tools available in PGC. As a core facility, we are able to prepare and load sequencing libraries for our clients in our sequencing machines. For example, in our National Biosurveillance Program, we are using three Illumina sequencing machines, the MySeq, NextSeq, and the NovaSeq 6000 platforms. From our array of different sequencing machines, the Illumina MySeq, NextSeq, and NovaSeq platforms are ideal choices because of their scalability and their capacity to deliver large volumes of data in a short amount of time, typically 3 to 5 days. In our program, the weekly goals are to generate at least 250 near complete genomes and then assign at least 350 COVID-19 samples to GSAID or WHO lineages. To achieve these targets, we multiplex or pool samples for processing in one sequencing experiment. For example, we process 750 samples in the NovaSeq, 350 samples in the NextSeq, and 25 samples in the MySeq. We are actually using PCR enrichment using publicly available Arctic primers to amplify at 2000x coverage all the regions covering the whole 30 kb genome of the SARS-CoV-2. To date, PGC has sequenced more than 25,000 samples to generate genomes from Philippine cases. Our current COVID-19 biosurveillance was first started through a proof of concept for COVID-19 sequencing. This was implemented as a PCHRD project called Biosurveillance of COVID-19 in the Philippines through whole genome sequencing of SARS-CoV-2 from patients. The project aimed to establish standardized protocols for COVID-19 sequencing and then to detect COVID-19 through sequencing of samples from collaborators. A lot has happened since then. We initially utilized shotgun metagenomic sequencing strategy to sequence and assemble the SARS-CoV-2 genome, but it has a low success rate. After we transitioned to amplicon sequencing using Arctic primers and the COVID-seq reagent test kit, our success rate has improved and used this strategy in our national COVID-19 biosurveillance program. Of course, other optimizations in our protocols were explored due to the diversity of the samples being enrolled and sequenced in this setup. One of our important findings is that longer read lengths improve our sequencing success rate, as shown in this figure. It's also important to note, however, that this setup has implications on our sequencing costs. Once our protocols have been finalized, complete implementation of our biosurveillance program with the DOH Epidemiology Bureau started. These are the different bulletins we released in 2020 and 2021 in relation to our COVID-19 sequencing. We highlight here the hallmarks of COVID-19 detection in the Philippines. In August 13, we released a bulletin discussing the detection of the very first notable variant of 2020 in our country, the D614 mutation. Most data now prove that this mutation was one of the main drivers of COVID-19 spread in the Philippines in 2020. Interestingly, PGC Visayas just recently detected a notable lineage or variant as COVID-19 rises again in our country in the same period as last year and 2 years ago. As more samples from the Philippines are sequenced and compared with samples from other parts of the world, we were able to do a more comprehensive comparison of SARS-CoV-2 genomes, which enabled us to infer three major possible routes of SARS-CoV-2 infection in the Philippines. The first possible route was through seafarers from the MV Diamond Princess because most of their genomes have very similar lineages to those coming from Japan. Other alternative routes could be, of course, foreign visitors from China and OFWs and travelers returning from Europe and the Middle East. This finding was one of the first scientific evidence to support tighter measures from incoming travelers. In January, we received our COVID-sick test kit reagents from our PCHRD project. We were able to sequence more samples from the Philippines. The optimized assay and sequencing run allowed us to improve our genome assembly rates so that we can analyze Philippine samples in more detail. This also allowed us to compare many samples from our country with worldwide SARS-CoV-2 data. As we see in the mutation analysis conducted in January 2021, as shown in the figure below, the most common hallmark mutation in most Philippine samples is still the D614G mutation, in contrast to most of the world, which was seeing a different set of mutations that were later categorized specifically to the UK and South African variants. Interestingly, a week later, the first case of the UK variant, now called the Delta variant, was detected in the Philippines. In this announcement paper, we pinpointed the index case to come from the UK, moving into the Cordillera Administrative Region, creating a cluster that's unique to the region, and independent of the transmission of the UK variant in the National Capital Region of the Philippines. Many more samples were sequenced in the following months, and we have started to look closely at specific mutations and candidates for hallmarks of a new variant. In March 13, we announced the detection of a novel lineage that started in the Philippines, particularly in Region 7. Shown here are the notable variants detected in the world in 2021. We were able to characterize the novel P.3 lineage after a comprehensive comparison of Philippine cases with worldwide variants or lineages. The P.3 lineage called the Theta variant was detected by PGC in January 2021. We continued to detect this P.3 variant in the Philippines for the rest of 2021, although it was not as widespread as the then dominating Delta variant. Moving to the current year, we know that most COVID-19 cases in the country have been assigned as Omicron variants. Through DOH announcements, we were detecting the following Omicron subvariants: the BA.2 variant in January, BA.4 in May, BA.5 in June, and then the BA.2.75 this month. These BA.2.75 cases were detected as the country deals with the fresh surge in infections driven by the Omicron BA.5. However, despite the rise in cases, the entire Philippines remains low risk for COVID-19 while Metro Manila is moderate risk for the virus. Looking forward, we'd like to go through this pandemic by successfully implementing our COVID-19 management measures based on the science. And the future of this field is bright because we now have the Philippine Genome Center and other agencies equipped with the capacity of doing genomic biosurveillance in conjunction with field epidemiology. As detection and management is similar to most genetic diseases, our current experience and capacity provides a great opportunity for us to embark on a sustainable and robust biosurveillance for future pandemics. If you'd like to collaborate with us on our current programs or avail of our sequencing services, you may visit us at UP Diliman or reach us via email or our social media accounts. Our core personnel of 8 members would be happy to correspond and work with you on your genomics work. Thank you very much for listening.
Oh, thank you so much for that quite a short presentation of the introduction about the PGC. So maybe we can entertain questions, and maybe you can talk a bit more about the maybe the current challenges that you have in PGC. Maybe that's my first question while we're waiting for others.
Okay. Actually, sir, we've heard a lot of challenges. So early in the pandemic, we were having trouble or difficulties with processing a lot of samples. So the first major hurdle was automation. So, but we were able to address that using, of course, machines that we've gathered or that were donated to PGC. So we now have a lot of automation machines such as the Kaya Cube, Kaya Connect, Kingfisher. So all of these were acquired through several donations from WHO, the DOH Epidemiology Bureau. Right now, I don't think there is many issues or constraints with regards to the biosurveillance. Um, but I think what remains since ever since is actually the sample collection. So of course, since we are collaborating and connecting with a lot of different hospitals and institutions, um, sample collection is going to be a bit more complex. So there will be different types of samples or sample quality. Then there will be sample logistics, what do you call this? Samples that are really having a hard time to go to PGC, and which is also good now. I think the good news is that we now have the PGC satellite facilities, particularly in Visayas and Mindanao, in the UP campuses in Visayas and Mindanao, to actually handle samples coming from the regions.
Thank you. We have a question from Dr. Raimundo.
Yeah. Before, when we had a project that required sequencing, we used the Malaysia facilities because they're cheaper than yours. Has that improved? Thank you.
Okay. Thank you, ma'am, for that question. No? We're actually working towards making our costs and prices much more accessible. I would say, at least for the standard capillary sequencing, it's still competitive. With regards to the NGS, we're doing our best to lower the prices. We're actually in close communication with our reagent suppliers. So if we have I think for now, the important thing is when we have a particular project, let's communicate po and do several consultations so that we can arrange a competitive pricing with you and with our reagent supplier. That's what I can say po.
Okay, thank you.
We have a question from Loida Perez. She asked, well, she wanted to know if there are other surveillance efforts in PGC on other emerging infectious diseases in the country that may be of concern to us.
Right now, at least PGC is not really the mandate of PGC does not really cover biosurveillance. I believe that's one of the major what do you call this, tasks of the RITM. But of course, we're working closely with RITM right now with regards to biosurveillance of the monkeypox virus. So we're developing our sequencing protocols again for monkeypox, and of course, right now RITM has established their own monkeypox RT-PCR detection. We're also trying to optimize our protocols for that, but we are focusing really on doing the sequencing the same way as we are doing for COVID-19 right now.
We have a question from Lisa Virata. She asked, what are the rapid antigen test kits available in the Philippines? Are they able to detect circulating Omicron variants BA.5 and 2.75?
I think there are a lot of antigen test kits available now. I'm not quite sure if there is an issue with regards to the detection of the BA.5 and BA.2.75 of these test kits. I'm sure, I think it's able to detect the BA.5, BA.2.75, that I'm not sure.
We have a question from Dr. Lydia Leonardo. Can you predict the virulence and the transmissibility of the variants that will emerge in the future? That's an interesting question.
Theoretically, yes, we can actually. That's what our experts are doing, and that's the reason why we're doing a lot of sequencing. So when we have the genetic information, we'll be able to predict the proteins, the coding regions of the virus, and then we'll be able to see the several functionalities of these proteins, of these molecules with regards to the virus, and then we'll be able to see if it will be more transmissible, it will be more pathogenic, or it will be of concern to the human host. So we are able to do that using several prediction models, bioinformatics analysis, for example, what do you call this, ligand, protein ligand interactions, all these things. So yun po.
Yeah, very good. So there's a question from Dr. Caroline Haraula. She asked, would you accept environmental samples for community wastewater-based epidemiology to complement your current clinical samples?
Yes, we do. Actually, a lot of studies in the US, for example, for COVID-19 biosurveillance are using wastewater. So it's a form of pulling samples together without having to collect from human samples, of course, and they are able to detect COVID-19 in a community using the wastewater. So I think for us, we've been doing or processing a lot of samples from water, for example, to look at the species diversity, for example, in water. So we are able to also do that, for example, in the context of wastewater to detect COVID-19. So we have several projects doing metagenomic sequencing from soils as well. So to see species diversity again of a particular soil of interest. And I think we've had several projects from MSI looking at analyzing the metagenome of water samples, for example, from seawater.
Dr. Leonardo asked, how do you compare BA.2.75 with other variants?
Recording in progress.
Right now, I haven't seen a more extensive study with regards to BA.5, but what makes it unique is that it has a hallmark mutation that's different from, at least that makes it unique from the original Omicron variant. For now, I think there is belief that it's highly transmissible.
And she follows that are they also highly virulent? Because they said there's warning about high virulence and transmissibility.
There is reason to believe so, but I think the studies have to be followed through to see. And I think right now, since there is increase in the COVID-19 cases in the world, again, that's being driven by this BA.2.75, then probably there is reason to believe that it's increasing transmissibility. With regards to virulence, that we're not sure, at least in the Philippine setting, we're increasing the COVID-19 cases, but hospitalizations, thankfully, is not as high as before.
In terms of others services that you have right now, there's of course a lot of COVID analysis. Are there any other samples that you've been receiving or in terms of sequencing these days or maybe before?
Oh, actually, we've been doing COVID-19 sequencing not only for the government through our biosurveillance, but also we're doing sequencing for clients. So, for example, we had services catered to several vaccine trials. So we had at least 3. So of course, I won't be able to give the details of this vaccine trial. So, but that's one of the things that we've been doing for COVID-19 sequencing. We have had also several collaborators in the hospitals that's interested in looking at how do you call this? More looking more comprehensively on the state of infection of COVID-19 in the hospital, trying to look for the source of infection. So we are able to do a bit of that. So at least we are able to pinpoint whether it's hospital acquired or if it's a community acquired COVID-19 in a closed hospital setting, something like that. So those are the things that we've been doing outside of the national biosurveillance. And of course, researchers and sequencing outside of COVID-19 sequencing as well. So like RNA sequencing for gene expression analysis of our health programs, things like that.
So there's a follow-up question here from Gerald Anthony. Aside from infectious diseases, can PGC accommodate molecular testing for cancer specimens? If so, is it possible to send paraffin sections, for example? Does PGC prefer purified nucleic acids? Because when he acquired last 2019, PGC is not yet allowed to process direct biological specimens, and he was advised to submit purified DNA instead. Is that maybe you can confirm?
Yes, that's right po. So previously, we haven't really optimized our protocols for extracting DNA from paraffin samples, FFPE samples. Right now, since we have established our clinical genomics laboratory and being how do you call this? Being rebranded as the clinical multiomics lab. So it's going to be launched, I guess, by the end of the year or early 2023. So we'll be able to also receive FFPE samples. We're also establishing our histopath lab to be able to do some tumor profiling work, some cancer genomics services to be offered. Right now, we're doing a proof of concept for how the cancer risk profiling for breast cancer. So we're collaborating closely with Dr. Eva Cochonco de la Paz to be able to sort of be able to compete with the services of, let's say, local services, for example, from Invite, so that we'll be able to capture clients who are sending abroad for cancer genomics testing. So hopefully we are able to launch this as early as before the year ends, but hopefully it is fully, we are done with our proof of concept sequencing, and we are completely done and able to accept samples before before the start of, or by the early 2023, at least for cancer risk testing, breast cancer risk testing. So usually that's blood, but of course later on, FFP samples will be much more readily accepted in our lab.
Excellent. Loida Perez asked, how much is the cost for RNA sequencing? Do you accept library preps too? Checking if there is a possibility to do collaboration work on RNA sequencing with PGC to reduce cost.
Yes, we are actually, for RNA sequencing, yes, we're doing many of those. Actually, we have several projects that we're doing currently, and we are also actually open to many kinds of collaborative setups. So, for example, if you are highly knowledgeable and you have trained lab personnel to do the library preps, then we're fine with having your libraries loaded directly into our machines, of course, with the assistance of our technicians. And then, of course, we're also happy to do the live preps for you. If you have your library prep kits and deliver those to us, if that's the setup that you want. So we're highly flexible with regards to these kinds of collaborative opportunities. So I think we can talk more about this through email or through Zoom outside of this webinar. So I'm looking forward to receiving email inquiries and these collaborative opportunities.
Dr. Giselle Concepcion, maybe you have a question.
Thank you, Mike, and congratulations, Benedict. Finally, you are giving the talk in PASE on your work, because I had been inviting you to give that talk for a long time. So congratulations, because now the PGC has a young team that consists of Benedict. I know he does the DNA sequencing. We have Francis Tablizo, who's doing bioinformatics with Benedict, and also Carlo Lapid, who's a computational guy. So we have a young team here, and we know they're doing great work, because our group with Roy Luis now I understand patronizes the PGC facilities and expertise with our bacterial 16S sequencing, as well as also our transcriptome analysis. For the longest time, we were sending samples to Macrogen for 16S, and relied on other sources for our transcriptome sequencing. This really opens up a lot of possibilities for us researchers, and especially those who are doing peptide sequencing, also available already at the Institute of Chem. And we have here with us Professor Emeritus L.C. Menes, who's worked so hard many years on conoidean peptides. And now we correlate this with our transcriptome from the conoidean samples. So Benedict, thank you for that. And I noticed that for COVID, you have your 2 publications already, finally, because for the longest time they were on Med Review. So now they're published. So that's very, very great. It really establishes the expertise, the capability of the PGC. Now, in connection with new viral infections that are now plaguing our country, at the NAST, I'm a part of the ad hoc committee on African swine fever in swine, and the avian flu that's now resulted in the culling of more than 1 million poultry heads. So I just want to know if you would be prepared for such sequencing of variants, because you're always mentioned at our meetings. Benedict, please tell us.
Yes, of course, ma'am. At least for African swine, I believe PGC Mindanao has been doing a lot of sequencing for African swine. So there is an available testing in PGC Mindanao. And then of course, with regards to actual sequencing of these viruses, avian flus, right now we haven't started doing the sequencing. But we'd be happy to connect with those who are actually have collecting samples from this, and then we'll do the sequencing.
Thanks, Benedict, for that. Because, well, Jeannie Montoya, PCHRD Director, Executive Director, is with us. And so is Bai of the DA. So I think we have to prepare also for avian flu. It's H5N6, not the H5N1. And it's coming in through the ducts that are migrating to our major marshlands like Candaba and other swamps in the country. So I hope we will perform this function of the PGC by being ahead of nationwide problems like avian flu and African swine fever. It's really affecting our poultry and our pig farmers. Thank you.
Yes, ma'am, noted po. We'll do this sequencing and connect with the responsible agencies, and as well as colleagues po at Department of Agriculture.
Yeah, so that's very good point. No, so I hope there are efforts also for PGC and doing that. There is a question from Dr. Maria Peña, and she asked if regarding about your standard protocol for sample preparation, do you have a SOP when standard protocol for sample preparation when sent for analysis in your core facility? Maybe you can elaborate a little bit on that.
Yes po. We have an SOP for sample receiving, as well as sample submission guidelines with regards to COVID-19 sequencing. I believe that's being distributed through the RITM to our different partner hospitals. So basically po, what we actually require from COVID-19 sequencing labs, collection labs, is that they have to have RT-PCR tests done, because we need the CT values, and we're only sequencing samples with a certain CT threshold of 20, 30 and below. I think 20 actually. That's because in most of our sequencing experiments, we have high success rate of generating high quality sequences from these samples. Beyond this threshold, we aren't able to generate good sequences, and so there won't be any lineage assignment. So I believe most of you are familiar with garbage in, garbage out in terms of molecular biology. So that's the thing that we're actually trying to mitigate, having processing and receiving samples that are high quality, so that we have data output that's also of high quality. So our comprehensive sample submission requirements, of course, for other samples outside of COVID-19 sequencing are available in our PGC website, DNA Sequencing Core Facility website. So of course, we also have those, so that we are able to deliver the results that our clients want with regards to their samples and our processing for sequences. So yun po.
I'd like to ask, you mentioned about the cost, for the next generation sequencing. That's where the country is trying to go towards, is the omics, of trying to do these sequences, but it's still kind of expensive. Do you have any maybe suggestion to an investigator, principal investigator, of what strategies they could do to lower the costs of doing this? I don't know, a pooling of the samples or what? Maybe you can have some suggestions.
Thank you, sir. That's always been our challenge, no, making genomics accessible and lowering the costs. Um, that's because also I think most of the reagents are being imported, no, so and that's something that's really hard to address, no, for now. But I think in our experience, we can sort of lower the costs by, of course, processing more samples, as most of our high throughput sequencers are highly scalable, and the more samples that you process, the cheaper the sequencing becomes per sample. So I guess at the top of my mind right now is to be able to find colleagues with similar applications and then combine the samples together, process them together, so that several projects can sort of um, make take advantage of lesser sequencing costs by having more samples processed together. Um, that's what we're trying to do at the center. However, there is not many research applications being done currently. But what I see in the future, now that we've actually had already have a genomics consortium, especially in the Visayas and Mindanao. I think having that conversation, consolidating samples with similar research applications or sequencing applications, it will be much more possible. And so taking advantage of the scalability of the sequencing machines will lower the cost, I think. That's our strategy for now. And we're doing our best really to look for other things to be able to make sequencing more accessible.
That's very good, no? So I think I like that idea as well. So the consortium, definitely, if more people are talking to each other and creating this program, and maybe more program projects with similar sequencing goals, no, that they're similar samples that can be run in high throughput. That would be very good. I don't see any other questions from the audience. But yes, Dr. Concepcion.
Yeah, perhaps Benedict can tell us the thrusts of the regional PGC facilities. So UPLB, of course, and then UP Mindanao, UP Visayas, because you have a publication on shrimp, studies. So just so the audience knows what you're working on currently in this, say, three CUs of UP involving non-UP establishment as well.
Okay, I think for now po, I'm not very familiar actually with most of the research trust, but I am familiar at least with the activities that's related to PGC in the satellite facilities. For example, in PGC in UPLB, there is a planned increase with regards to bioinformatics, just because there is an arrangement with an expansion of the bioinformatics center in PGC Agriculture in UPLB. And then in PGC Visayas, of course, they're still focusing most of their work on the sustainability of the PGC satellite facility. I think most of the projects that are anchored to the DOST funded expansion to the regions with regards to establishing the PGC are mostly marine and aquatic sequencing. So that has applications on diversity. I think that's mostly for PGC Visayas, and then for PGC Mindanao, a lot of sequencing projects are actually focused on biosurveillance actually. So we have a big project in PGC Mindanao, UP Mindanao on monitoring coronaviruses in bats. So and I think that's going towards the One Health. No, so in general, PGC is embarking on a One Health approach to biosurveillance. So we're not only looking into sequencing surveillance of viruses in the human populations, but as well as our animal reservoirs. So I'm not quite confident po with regards to these updates on the PG satellite facilities. I hope yung mga colleagues po natin from PGC satellite facilities are pasensya na po, parang ganun.
So
But that's what I can say.
Thank you so much for that. Just a follow up to that question. So if let's say an investigator would like to study to do some sequencing from aquaculture, for example, is it better to send those samples to PGC Visayas even though they're in Luzon, or will PGC Maine accommodate those as well, or maybe you can have some comments on that?
That's a very nice question, sir. So actually, the satellite facilities are not there to focus on several things or to be a competitor to PGC in what you call this, in Manila or in Quezon City. But I think the satellite facilities are there for accessibility. So our satellite facilities in Visayas and Mindanao have their sequencers, medium throughput sequencers. So the client can actually send to the PGC that they want to send to. But of course, if it's not available, for example, they require a much higher throughput. Of course, we have those machines in PGC UP Diliman. But if for example shipping is a problem now and they're in Visayas, then I would suggest that they contact and connect with PGC Visayas, for example, or PGC Mindanao. So actually that's our vision po and our goal, for the sequencing services to be available in their regions, in their local regions, so that our scientists are not bothered with the hurdles and the challenges of sending samples through shipments, no, through shipping. So yun po kasi usually, di ba, yung problems, especially now that, for example, RNA and DNA extracts are more or less what you call this, affected by shipping. So we'd like to preserve the integrity of these samples, and so having those sequencing facilities accessible in your area is really important.
Thank you. Dr. Giselle Concepcion, do you still have any questions?
I think it's important, Benedict, to identify the persons in the UP CUs to contact and who are the experts there. For example, the new UP Mindanao Chancellor Lara Nerao, she's in the PGC, right? And then we know those in UP Visayas also. Now, in particular, we want to find a place where the northern part of the country, northern Luzon, perhaps can get these sequences done, because many of the farms, say for poultry and for swine, is starting in Luzon. And we know that bird flu will probably spread because of the Candaba marsh swamps. No, so we're talking about northern Luzon. Of course, CLSU is there, but then you have UP Baguio, uh, in the Cordillera area. Also, maybe Mindanao State University. Are there plans for the PGC to get leading SUCs in the country involved para hindi rin lang lahat UP? Kasi we don't have a presence in many parts of the country where this kind of sequencing is needed.
Actually, ma'am, that's been in the talks previously with regards to do we need a PGC in North Luzon. But at least for now, the PGC in Quezon City is available for most of the areas in Luzon. Of course, we've been to note po, we've been collaborating with a lot of universities and institutes in Northern Luzon. So for example, we've been doing several projects with CLSU, with PCC, and of course Benguet State University, and a lot of other universities po that I'm sorry, I can't remember now. And then of course, with regards to contact persons po, no, for PGC Visayas, our program director there is Dr. Noel Ferriols. And then of course for PGC Mindanao, our previous program director is actually Chancellor Lair Morao. Now we have the program director now is Dr. Joel Tolentino. I hope I'm not mistaken. So you can actually look for our contact details in the respective PGC websites. Ayun po. And then for PGC Visayas, for agriculture in UPLB, we also have Dr. Peewee there as the contact person. Yes, ayun po muna. There is a comment po from Dr. Concepcion. Okay. Dr. Joyce Ibana po has strong links with Pampanga State University. Yes po, that's good to note po. Will be if they have needs po and opportunities for collaborations, we'll be happy to accommodate.
There's also a comment from Dr. Wengigia, and she asked, she comments about networking. So maybe you can tap to the DA Biotech program, not just the OST, especially for these funding for collaboration in terms of the services of PGC. And the biotech facilities under the DA Biotech program are just being built and upgraded. So maybe you should look into that as well.
Oh, thank you for that, Ma'am Yang. Actually, we recently had a meeting with PIDEA. So it's a government agency with regards to biodiversity barcoding of what do you call this? Of trumpet plants. So, and one thing that came to mind is actually to apply for a grant in the DA Biotech, because most of the calls from the OST now has ended, no, so, and hopefully we are able to access the grants from the DA Biotech. I know that they are also able to provide and enable genomics projects. So yun po. Thank you po, Dr. Wayne.
Thank you very much. And it's already 8:55, and I think we have a very, very good discussion today regarding activities being done at PGC and the services that they can offer us. And I hope that each one of us may consider going to PGC. Hopefully, if we have a consortium, if we collaborate with each other and have more samples to be analyzed in PGC, it will bring down the cost even more. And then now we can do a lot more activities here in the Philippines. If you think about it, no, especially if the funding is coming from the Philippine government and the money is just circulating within the Philippines and not we're not purchasing services abroad, it also is benefiting the whole Philippines at the end in the long run. So I hope we can do that and then strengthen our activity and research efforts in the Philippines. And thank you very much for this. If you have any questions and further concerns, and if you want to collaborate with PGC, feel free to check the PGC website and contact Dr. Ben Meralit for all your sequencing needs. Thank you very much for this talk. And there's a comment, just last comment, picture plans of interest to Mindanao researchers. It's a great potential for research collaboration also. And CMU's Victor Amoroso and Ann Villarobos, the transcriptal analysis of hydrolytic enzymes. So thank you very much to everyone, and I will leave at this. Thank you so much for attending.